Okay, everyone, we're back now with part 2 of the video lecture for chapter 13. You'll recall at the end of part 1 of this video lecture, we looked at the effect of variable environmental parameters or conditions and how they can affect the competitive ability of different species. We looked at the example here with the brook trout, brown trout, and the creek chub, and how their competitive abilities changed with water temperature. Okay, so now we're going to kind of, along those lines, begin to talk about how it is that variation in environmental conditions over time can affect competitive interactions. So a species that is more efficient at exploiting a shared limiting resource may exclude other species. But when environmental conditions are variable over time, the competitive advantages of species may change. One species may be at an advantage under one set of temperatures, let's say, and another species has more of an advantage under other temperatures, water temperature, air temperature, whatever. So if the change then in the environment is continuous, no one species may reach sufficient density to displace its competitors because no one species is always the best competitor. Environmental variation allows competitors to coexist when one would exclude the other under constant conditions. It allows more species to exist in a given area, basically. So a number of studies have looked at this sort of thing. And so, for example, there was a study of grass species living in a savanna community in southwest Zimbabwe in Africa. And over a 10-year period, the dominant grass species shifted from Eurocloa mozambicensis, mouthful, to Heteropogon contortus. So the question is, what caused this shift? And here you can see the shift. So here we have, the year along the x-axis and the percent occurrence, okay? So you see that Heteropogon, for example, here in the purple line, was a very low occurrence initially over these first few years from, say, 1971 to 73, 74, but then began to increase quite a lot after that. You notice that initially, Eurocloa was most abundant, hit a peak in 72, 73, then gradually declined after that. So the shift is in response to changing patterns of precipitation during the same period, which we'll see in a minute. Eurocloa was able to compete successfully under the drier conditions during the 1971, 72, and then 72, 73 rainy seasons. So that's what you're seeing right here, Eurocloa being the better competitor under these drier conditions. We'll see the precip figure here in a minute. With the increase in rainfall that began in around 1973, 74, Heteropogon began to do better because it does better under wetter conditions. All right, so this shift then was a result of yearly variations in rainfall. And again, Eurocloa can survive and grow better in dry conditions. Heteropogon does better under wetter conditions. And this is a graph showing you the precipitation patterns for those years. You see that 71, 72, 73, 74 were fairly dry years. Then we had an increase in precipitation, and that's the point where Heteropogon began to dominate and where Eurocloa began to drop, if you recall. We can go back and look at that again real quick. And so you imagine that we have the precipitation being very, very little right here. For example, you have some dry years, and that's why Eurocloa, that does better under dry conditions, did so well then. But then as our precipitation went up, you see that Eurocloa began to decline and Heteropogon began to increase. And again, there you see it. So that's an example of how changing environmental conditions can affect the competitive ability and the interactions between different species. Now, climate can also have a density-independent effect on population growth. Periods of extremes, such as drought, flooding, or extreme temperatures, hot or cold, may reduce population size below carrying capacity. If that happens, frequently relative to the time needed for recovery, or in other words, for a population to reach carrying capacity, resources may be abundant enough at any given time to reduce or maybe even eliminate competition. So population size is an important factor as well. Competition between species often involves multiple resources also, And so competition for one resource could affect an organism's ability to use other resources. All right, some examples. Many birds in temperate and tropical communities exhibit interspecific territoriality. Some species defend territories against closely related species. Other species, however, may have to defend territories against a broad range of competitors. It just depends on what the situation happens to be. In plants, there are many examples showing how competition for one resource can influence the ability of an individual to exploit other resources, and that can affect growth and survival. Both greenhouse and field experiments have been performed that show this very thing. As an example of a greenhouse experiment, some researchers looked at the competition between subterranean clover and a species of plant called skeleton weed. And this is the basic experimental setup right here. So plants were grown in the greenhouse in, first of all, monocultures. So on the left, we have skeleton weed grown alone. That's a monoculture. We have subterranean clover grown alone as a monoculture. Okay. So they were grown in pots, so the roots and the leaves intermingled. Now, the mixture population. So we have two populations here. A couple of variations on this. You have two populations grown in the same pot where the leaves and roots intermingled. Okay, so here we have, we have two populations grown in the same pot where leaves and roots intermingled. We have them grown in the same pot where roots intermingled but leaves were separate. So here you have, for example, roots being able to intermingle, but the leaves are separated by some sort of barrier, maybe a plastic plate or something like that. Then we have them grown in separate pots where the leaves were actually capable of intermingling, but the roots were actually separated. Okay, so and then you had control species as well that were grown in separate pots entirely. So the effects of competition were determined for both above ground resources, and in plants that's going to be light primarily, and also below ground resources, which would be water and nutrients. And so what they found, or the results rather, showed that clover was not significantly affected by the presence of skeleton weed, but skeleton weed was very much affected adversely under all the conditions when the plants were grown together. So the question is, what does that tell us about resource competition between these two species? Well, it says the species are competing for both above ground and below ground resources. They're not just competing for one resource, they're competing for an entire suite of resources. And compared to skeleton weed grown in monoculture by itself, when the two species were grown together, effects on skeleton weed biomass were pretty dramatic. When the roots were allowed to intermingle, skeleton weed biomass was reduced by 35%. So that's what you see right here. Okay, so skeleton weed biomass was reduced by 35%. When the canopies were allowed to intermingle here, skeleton weed biomass was reduced by 53%. And when both roots and canopy were allowed to intermingle in the same pot, you had skeleton weed biomass reduced by 69%. You can see how small they've drawn the plants there to sort of show that. So this means that clover or subterranean clover plants were superior competitors for light, water, and nutrients. They were superior competitors across the board for all these resources. All right. Another example, field studies in an old field grassland community in Pennsylvania have been conducted, and the experimental design here was similar to a greenhouse study. What they did is they used PVC root exclusion tubes to control root interaction by varying the number of holes drilled in the tube. So the idea is you plant a plant in the soil and you encase its root ball or root structure in a PVC tube. If there are no holes in that, there's no way for roots of neighboring competitor plants to interact with the roots of the plant in question, and you would get no competition. But if you drill holes in that PVC tube, then there are avenues through which the roots of a neighboring competitor plant could intermingle with the roots of the plant in the tube, and then you would get competition. So They tied back the neighboring vegetation to vary the light level and different treatments for light level. They created 16 combinations of above and below ground competition. So again, above ground looking mostly at light, below ground looking at water and nutrient availability. Control plants were completely isolated from neighboring plants, so no competition would be going on there. And this is essentially what you see. So first of all, here's a PVC root exclusion tube. You see some holes in it, right? There's a plant sort of growing in that. And so you can imagine if there are holes, then these neighboring grasses and things would have a way for the roots to get in and compete with the roots of the plant that's actually inside that tube. All right, so the first figure here, we have the percent of tube open to neighbors. So 0%, that's a tube with no holes drilled in it, all the way up to a tube that apparently, or maybe even no tube at all in the case of 100% open to neighbors, 50% open, 25, and so on. So from left to right then we have increasing root competition. On the y-axis we have the neighbor root biomass. So the amount of biomass of neighboring roots found inside the tube at the end of the experiment. And you can see that as they had more access, the neighboring root biomass inside the tube increased. And so as competition went up, you had neighboring root biomass going up along with it. Now, what effect did this have on the growth of the plant inside the tube? That guy there. That's what this figure shows. So again, percent of tube open to neighbors, but now you see the target plant biomass. Our target plant is the one inside the tube. Highest biomass was when neighboring roots could not get into the tube and compete. And then, so again, as our competition goes up, we have decreasing plant growth, okay? All right. So the results then show a clear pattern of interaction between above ground and below ground competition. The increased competition for below ground resources, so water, soil nutrients, that kind of thing, reduces growth rate and plant height, and then that reduction leads to a reduction in competitive ability for light. Okay, so the two actually interact with each other. The above ground set and below ground set of resources can interact and affect one another. Or competition for one can affect competition for another, let's say. All right, so as environmental conditions change, the relative competitive abilities of species change. We saw that with the fish, for example, right, the trout. But the question is, what can cause these shifts in competitive ability? Well, shifts in competitive ability can result from, first of all, changes in carrying capacity of the population in question, so changes in K, all right, and changes in the physical environment. All right, so changes in carrying capacity for a species because of a change in the resource base, for example, and then secondly, changes in the physical environment that interact with resource availability. So these changes in competitive ability along some environmental gradient, like temperature, for example, have been examined for both plants and animals. Greenhouse studies in Australia have investigated the changes in interspecific competition between plant species across experimental gradients of nutrient availability. One study in particular examined 6 different species of thistle plant. The gradient was established by applying a nutrient solution. So they're looking at a gradient in soil nutrient concentration here. So plants were grown in monoculture of single species and in a mixture of all 6 species grown together. 11 nutrient treatments ranging from 1/64th to 16 times the recommended concentration of some standard nutrient solution were applied. And after 14 weeks, the plants were harvested and their dry weights obtained to get a measure of biomass. All right, so plants grown in mixed cultures showed much variation, and the response was different than the response was for those that were grown in monoculture. Interspecific competition affected growth for each species, and the competitive abilities changed along the gradient. And that's what you see right here. So what you're seeing then is, first of all, the different species, 6 different species, okay? And you see how their total biomass changed along our gradient then of relative nutrient concentration. all of them grew less and had less biomass at low nutrient concentration compared to high. And overall, most of them had the same sort of S-shaped curve or pattern of change in biomass with nutrient concentration. However, what you want to look for is where the lines intersect or cross. So initially, if you look, we had Carthamus lanatus right here as the best competitor initially at low nutrient concentration, but then right there we have Cardus pinocephalus that begins to outcompete it at mid-level nutrient concentrations, and it does well up until this point where Silybum marianum begins to outcompete it, okay? So I'll explain why I have these arrows highlighting those three species here in just a moment. All right, so if we examine the relative response of species in the mixed species experiment, we find something very interesting. If, for example, you divide the biomass value for a particular species at a given nutrient level by the biomass value of the species that has the highest biomass at that nutrient level, in other words, the best competitor, this gives us something called the normalized ecological performance. So, normalized ecological performance. All right. So, for example, if we divide the biomass of Cardus nutans right here, say at 1/64th, by the biomass of the best competitor, which in this case was Carthamus lanatus, then we get a number of less than 1. So if that's what, about 2 biomass, and this is about 5, so that'd be about 0.4, 0.4, that gives us a relative measure of the competitive of that species at that nutrient level. That is the normalized ecological performance. Okay, 1 would be the highest, the best competitor. All others must have a number less than 1. Okay, so what we find then, by looking at the relative responses then through normalized ecological performance, we see that the dominant thistle species in the experiment along the nutrient gradient, which was Silybum marianum, is the best competitor at highest nutrient concentrations. Okay, at highest nutrient concentrations. So if you look over here, high nutrient concentrations, here we have Silybum marianum, which is doing the best. So if we actually plot that, okay, our normalized ecological performance of these three species, the ones I had shown with the arrows over here just as an example, what we see is this, right? So you see that at very low nutrient concentration, we have Lanatus doing very well, but then as nutrient concentrations go up, it begins to drop off in terms of its growth rate. Very often, high concentrations of things that you might think would be good can turn out to be bad. Too much oxygen can kill you if you have it in high enough concentrations, for example. But then, early on, we see that Pinocephalus, Cardus pinocephalus, starts off as really the worst competitor at low nutrient concentrations, but then it picks up, does really well over a fairly broad range of nutrient concentrations, 1/16 up to about 4, but then nutrient concentrations go too high for it and it begins to decline. If you look at Silybum marianum, it starts off as a little bit better competitor than Pinocephalus, and then sort of picks up gradually along the way and ultimately outcompetes both of those species. And if you look at these points where they intersect and compare them with what you see here on the graph, for example, right there and right there, that matches up very nicely with what we see here. Okay, this is just another way. The normalized ecological performance is just a way to compare species competitive ability more directly. All right, so I think that is probably going to be a good place to stop for this. This is going to be a little bit shorter than a half hour, and I think the next one probably will as well. But that's sort of a natural stopping point for this particular section of the chapter. So we'll stop here and pick up with the next topic in part 3 of this video lecture for chapter 13.